sequence of amino acid in a protein is determined by genetic code. AUG, methionine, and start codon. Coded by methionine, one code, degenerate. Increase in number of genetic code, tryptophan, phenylalanine, valine, leucine. mRNA, direct building of protein through sequence of codon. Genetic code, it has 61 codon for amino acid and 3 stop codons. It is universal and has 3 bases per codon. Some amino acids are coded by multiple codons, non-ambiguous. UCA, UU, UUCC, GGG, AUG, AGU, serine, phenylalanine, serine, glycine, serine. Experiment cracked the genetic code Nirenberg and Mathai. Universal code for phenylalanine UUU. UUU phenylalanine. Triple G glycine. UCU serine. Triple C proline. AUG methionine. Addition and deletion, frame shift mutation. Codon, presence of mRNA. Anticodon, adaptive molecule tRNA. Codon not have tRNA, stop codons. Sequence of amino acid and polypeptide determined by mRNA. Transition of mRNA, ribosomes. Translation is related by protein synthesis. Smaller subunit. Amino acylation of tRNA, formation of peptide bond. Peptide bond during translation is affected by ribosome. Inducer lactose beta galactosidase, lag on which allactose binds to repressor protein. Lactose function as inducer that binds to repressor protein. Inducer regulate on and off. RNA polymerase has access to promote and transcription process which the repressor inactivate. In absence of lactose, repressor binds to operator region. Lacoperon, Jacob monode. A is transcriptase, I is repressor protein, Y is permease, Z is beta galactosidase. Lacoperon consists of one regulator gene and 3 structural genes. High codon of lactose induces a molecule binds to repressor protein and RNA polymerase binds to the promoter sequence. Not part of transcription unit, inducer. DNA sequence based on Frederick and Sanger. Vector for human genome sequence, BAC-YAC. Aim HGP, identify the sequence of gene present in human DNA 1990. Initiation HGP in 1990. HGP, less than 2% of genetic genome codes for protein. Y has the fewest number of genes. SNP helps in the tracing human history. High frequency DNA polymorphism. Most number of genes, chromosomal number 1. Largest gene in the human, dystrophin. Single base DNA difference, SNP. Basis of DNA fingerprinting is polymorphic sequence. Father of DNA fingerprinting, Jeffrey. Southern blotting, transfer DNA fragments, amino acid into nitrocellulose sheet. Radioactive molecule, probe. Satellite DNA, forensic science. VNTR, DNA fingerprinting. DNA fingerprinting analysis made of VNTR. Technique of DNA fingerprinting, repetitive DNA, mini satellite DNA. Probe, known single stranded labeled DNA. Sequence, isolation of DNA, digestion of DNA by restriction endonuclease, separation of DNA fragments by electrophoresis, blotting, hybridization using labeled VNTR probe, hybridization DNA fragments by autoradiography, DNA fingerprinting, digestion of DNA by restriction endonuclease, electrophoresis of DNA fragments, blotting of DNA fragments to nitrocellulose membrane, hybridization with DNA probe, autoradiography. DNA satellite is important because high degree polymorphism and same degree of polymorphism to child. DNA fingerprinting, PCR based forensic science test for paternity. Probe, VNTR. DNA fingerprinting requires restriction endonuclease. Gene expression, gene transcription, mRNA translation protein. Operon, polycystonic structural gene regulation. Operon, binding to repressor protein. Promoter binds to RNA polymerase. Repressor, inactive by inductor. Definition: GTP dependent release factor. Translation: amino acyl tRNA synthesis. Transcription: RNA polymerase. DNA replication: Okazaki fragments. RNA polymerase binds to promoter and initiate transcription. Taylor and colleagues: DNA replication semi-conservatively. Hersey and Chase: DNA is genetic material. Griffith: transcription principle. Jacob: lag operon. Promoter: site for binding of RNA polymerase. Repressor protein binds to operator to stop enzyme synthesis. Operon: structural gene operon and promoter, not transcribed by DNA.